Okay, a couple things about the structure. First, they're a unicellular organism. That means that they have one cell. They have no nucleus, which is different than your eukaryotic cells. They have DNA. It's just not in a nucleus. Okay, and there's two parts of their DNA. One is their main circular chromosome. Okay, and it's not a perfect circle. It just means that it's in a loop. Okay, so that's their main DNA. And then they also have a little tiny thing called a plasmid, okay, which is a circular part of DNA. And this is the part that usually has genes on it for being resistant to some kind of drug or whatever medication that they are given for that. So single-celled, no nucleus, and but they do have DNA. As far as classification goes, this is the main thing. Prokaryotic cells do not have a nucleus. That's a pretty standard definition of a prokaryotic cell. Simple definition, but that's the basic one. Okay. This one, you know, there's a bunch of parts listed here, and you can kind of look at your coloring sheet for what they look like. Obviously, there's a picture here, but these are the parts they have. On the next slide, I'm going to go over what the functions are of just the ones that are different than we've talked about before. So the cell membrane and cell wall, which prokaryotes have, has the same basic function as in a eukaryotic cell. Now some prokaryotes or bacteria have a capsule, and that's a slimy thing that basically protects the bacteria from getting killed by your immune system. Okay, ribosomes, they have same function. DNA is the same function, stores information, has genes on it, replicates, all those things. They have these little things called pili, which are like little hairs, and they stick to other cells, and sometimes they're modified, and it's like a tube that leads to another bacteria, and they can exchange little parts of DNA there. They can actually exchange genetic information through that little tube. And then some have little tails, little flagella. They're like little tails, and that helps them swim around a little bit. Okay, here's some examples of conjugation, and that was the little pili that are attached. And this is how they exchange genetic information through this little tube right there, that little tube right there, that little tube right there. And yeah, they're happy. Okay. By the way, conjugation is another way of saying bacteria sex. So you sickos are looking at pictures of bacteria sex. Way to go. Okay. They're classified in different ways based on their shape. Now these things are tiny. Okay, so they're a lot smaller than your cells, than your skin cells and blood cells and so on. Really, really, really small. And I'll give you a to scale thing, a little animation in a little bit for that. And okay, here's that animation I promised you later. We're doing it now. I think we've seen this in class actually. It's just starting with the size of a coffee bean. We kind of zoom in and we go all the way down past the amoeba and the paramecium and the egg and the sperm and the skin and the red blood cell and so on till we get to the E. coli bacterium. Okay, so it's roughly the size of your mitochondria, which is inside of your cells. So it's a lot smaller than your cells. If we keep going, there are the viruses and the ribosomes and so on, all the way down to a carbon. Okay, so there you go. Tiny, 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 tiny little feller. And they also come in three basic shapes. Okay, that is rod-shaped, they are round or spherical shaped, and they are spirals. Okay, so those are the three main shapes of bacteria, and they're arranged in different ways as well. Okay, so they are found in clumps, and the prefix staphylo refers to the fact that they're found in clumps, and they're also found in chains, and the prefix strepto means that they're found in chains. Okay, and what those things look like, those are chains. Okay, so it's long chains of spherical bacteria. Okay, so there's a name for that. There's a prefix for the arrangement, and there's a word for the shape. So that would be called the Streptococcus bacteria. Incidentally, that's the arrangement that causes strep throat. That's why they call it strep throat. Okay, there's another arrangement, would be long chains of rod-shaped bacteria. Streptobacillus. Strepto meaning chain, bacillus meaning rod-shaped. And then there's clumps of spherical bacteria. The prefix is staph, so Staphylococcus are clumps of spherical bacteria. Again, staph infection, that's what we're talking about. Your coloring sheet at this point would be good to look at. It gives you those things, and you can color those, and it gives you just better pictures of that as well. I'm pausing here because we're taking a quiz right now. Okay, so that was pretty much one section. The next section is, so you may want to summarize that section, or some way in your notes say, okay, that was the structure and function of bacteria. How would I summarize that? Maybe pause the video and try to do that, and maybe do a little coloring as well. Okay, the next section, growth and reproduction. So basically, when a bacteria or a prokaryote gets too big, they just split in half. Okay, it's called binary fission. So basically that means that their DNA replicates and then they split in half. Okay, a little really detailed process right there. Okay, I hope it really clears it up for you. But it's asexual reproduction, meaning they're going to be identical, and they just split in half. And I think we'll get to it in a later slide. But when conditions are ideal, certain bacteria can reproduce every 20 minutes. If conditions are not ideal, sometimes they can collapse in on themselves and form what's called an endospore here, and that helps them survive conditions that are super harsh. And then when conditions are favorable again, they'll start to reproduce. And then again, the conjugation, which is the little transfer of genetic information through these tubes, and that's basically how they communicate about resistance to certain drugs. They can pass the genes through that little tube right there. Okay. So this one, every day I'm doubling. Most bacteria can do that in 24 hours, but sometimes, like I said, they'll reproduce every 20 minutes. So if you're a math guy or girl, if we start with just one bacterium, how many will we have after 2 hours? And how many will we have after 8 hours? Okay, you can just come up and tell me the answer and see if you can get it. Mr. Carlson will give you a treat. Okay. Now the disease-causing issue, the little thing there, less than 1%. Okay, less than 1% of bacteria actually cause disease. The remaining 99 are not necessarily bad for you. Okay, so there's two ways that they do it, and that is either by infection or toxin production. So if you get infected with one, they'll get inside of your body and they'll start to divide and divide and divide. They'll outcompete your cells for nutrients and oxygen, and they'll start killing the surrounding cells. So that's one way. And then another one is that they actually produce toxins that can be super deadly and make you really sick and kill you. So refreshing note. And a lot of times they do occur at the same time. And these are just some examples: Salmonella, Black Death, E. coli, botulism, and so on. Just some examples of diseases, and we'll do a separate thing in class where we go over some of the specifics of that one. Okay, let's move along. Some good things about them. So most of them are good. As we talked in our ecology unit, they are decomposers. The algae produces oxygen. They fix nitrogen so that the plants can use it and get that into the ecosystem. So a lot of that stuff we talked about already, and those are good things that bacteria do. For people, they're all over in your intestines. They help you break down all the food that you eat. They make some vitamins for you. They outcompete the bad bacteria. They make our food, some yogurt and cheese, etc. Industrial companies use them to break down food. Oil spills, they can digest oil spills, and then they actually are being attempted to use to make drugs to kill other bacteria. So lots and lots and lots of good things from these bacteria. The medicines and the drugs that we use to kill them are called antibiotics, and they do it in a couple different ways, and we'll talk about this during our lab. But They either kill the bacteria or they just stop them from reproducing. Okay. I think that might be it. A couple things to consider. These questions here, and you can look through, and we'll talk about some of the first two. But really, like if someone asked you what a prokaryote was, like could you tell them? And so hopefully, like by watching this, and you should be able to do that. If you can't, you might want to go back and watch it again. Okay, thanks. At this point, just click anywhere on the screen, and it'll take you to a Google form, and then input 2 questions that you have over the content.